Welcome to this explainer. Today, we're diving straight into the very core of what makes you, well, you. We're looking at DNA. It's basically our biological blueprint, and we're going to explore exactly what happens when those vital instructions change. You know, genetics is all about the study of heredity, and really, the whole shebang comes down to this microscopic code written inside ourselves. But what actually happens when that code gets scrambled? Well, let's find out. So here's our quick roadmap for today. 1. The genetic blueprint. 2. The types and causes of mutations. 3. Classifying genetic disorders. 4. Detecting those anomalies. And finally, 5. Treating and editing DNA. Let's get into it. Starting off with Section 1, the genetic blueprint, focusing on DNA and genes. Think of DNA, or deoxyribonucleic acid if we're being formal, as the ultimate architectural master plan. It literally contains all the genes that dictate the structure and function of every single cell in your body. It's incredible. Now, when there's a permanent change in the nucleotide sequence of that DNA, we call that a mutation. I know, mutation can sometimes sound like a scary sci-fi word, right? But honestly, it's an absolutely crucial source of genetic variation. It plays a massive role in evolution. But on the flip side of that, whenever there's an alteration in the sequence, it can absolutely interfere with our normal biological processes. Moving to Section 2, types and causes of mutations, looking at their origins. Let's look at the different ways this master blueprint can actually be miswritten, misread, or physically damaged. Let's break down these microscopic typos. Just imagine you're typing out a long text message. A point mutation is kind of like swapping just one single letter for another. Sometimes it doesn't change the meaning at all, but sometimes it ruins the word entirely. Then we have insertion and deletion mutations. This is where you either add an extra letter or just delete one altogether. And this doesn't just mess up one word. It literally shifts the entire reading frame of our genes. That can completely disrupt how our cells synthesize vital proteins. But, sometimes the error isn't just a tiny typo in a single word. Chromosomal mutations are massive structural failures. Think of this as entire paragraphs or even whole pages of your blueprint being ripped out, duplicated, or literally glued into the wrong chapter. These are your deletions, inversions, and translocations. And then we have numerical abnormalities. That's when there's an abnormal number of chromosomes altogether, like having an extra chromosome, which is known as a trisomy, or missing one completely, which is called a monosomy. So where do these errors actually come from? Well, they originate in two main ways. First up, spontaneous mutations. These are just natural errors. Every single time your cells divide, they have to copy billions of letters of DNA. Naturally, the cellular machinery is going to occasionally make a mistake during that replication process. Second, we have induced mutations. These are caused by harsh external elements in our environment that actively attack and damage the DNA structure. And what are those external elements? Well, our DNA is constantly battling everyday hazards. We're talking ultraviolet radiation from the sun, ionizing radiation, chemical mutagens, and even viral infections, oxidative stress, and environmental pollutants. Now, our cells are incredibly smart. They possess these amazing natural repair mechanisms to fix this damage. But the reality is, sometimes those induced errors just escape correction, and when they do, they become a permanent part of the genetic code. So this brings us to a really crucial question. What happens when the blueprint goes wrong? What actually happens when these permanent typos escape our cellular proofreaders and manifest physically in our bodies? That brings us to Section 3, classifying genetic disorders. When a mutation disrupts normal cellular function to the point of actually causing disease, we call it a genetic disorder. There are three primary classifications of these disorders to wrap our heads around. First, single gene disorders. As the name suggests, these result from a mutation in just one specific gene. Classic examples here include cystic fibrosis or sickle cell disease. Second are chromosomal disorders, which we touched on earlier. These are caused by those structural or numerical abnormalities, such as Down syndrome. And third, we have multifactorial disorders. These are incredibly complex because they arise from interactions between multiple genes and environmental factors. Think of conditions like type 2 diabetes, coronary artery disease, or Alzheimer's. Now, the impact of these disorders varies wildly from person to person. While some folks might only experience really mild symptoms, others face severe challenges. The effects can include developmental abnormalities, intellectual disabilities, metabolic dysfunction, where the body literally can't process nutrients correctly, severe immune system defects, neurological impairments, and, in some cases, a significantly reduced life expectancy. Let's look at Section 4, detecting the anomalies. I know it all sounds a bit daunting, but fortunately, we're living in a true golden age of molecular genetics. Modern biotechnology has given us some unbelievable new tools to read the human blueprint and spot these anomalies early on. Here are just a few of the most powerful diagnostic tools in a modern geneticist's toolkit. We use karyotyping to look at the whole chromosome picture, polymerase chain reaction, or PCR, to amplify very specific DNA segments, and incredibly detailed whole genome sequencing. We also heavily rely on prenatal genetic testing and newborn screening programs. Why? Because early detection is absolutely critical. Spotting a mutation early on enables timely treatment and drastically better disease management for the patient. Which leads us perfectly into Section 5, treating and editing DNA. Because once we actually detect a genetic anomaly, how do scientists and doctors go about fixing a house that was already built with a faulty blueprint? The modern pipeline of patient care essentially moves logically through these 3 steps. Step 1 is early diagnosis, identifying the exact genetic mutation. Step 2 is treatment management, using specific therapies to improve the patient's immediate health and outcomes. And step 3? That's the cutting-edge frontier, genome editing, where we actually try to go in and correct the DNA itself. Let's look a bit closer at step 2. The reality right now is that many genetic disorders simply cannot be completely cured. However, we have highly effective ways to manage them. We're talking about enzyme replacement therapy, targeted drug therapies, specialized dietary management, physical rehabilitation, and really vital genetic counseling. These diverse treatment approaches significantly improve a patient's daily quality of life. But step 3? That is absolutely where the future lies. Enter CRISPR-Cas9. This is a truly revolutionary genome editing technology. Instead of just managing the symptoms of a mutation, CRISPR offers the opportunity to go straight to the source. Scientists are developing personalized medicine approaches to literally act as molecular scissors, cutting out the faulty parts of the human blueprint and replacing them with the correct genetic code. It is an astonishing, real game changer for healthcare. I want to leave you with this final thought. While we must absolutely use biotechnology to treat genetic diseases and reduce human suffering, we have to remember the dual nature of what we're looking at here. Mutations are fundamental biological events that generate genetic diversity and drive evolution. Every single difference in our species started as a tiny typo in the dark of a cell. As we gain the power to rewrite our own blueprint, the question isn't just about what diseases we will cure, but how will we choose to shape the future evolution of humanity itself. Thanks for joining me on this explainer, and keep questioning the code.